Chandrasekhar Venkat Raman. It was on this day, in the year 1928, that he discovered the phenomenon of the scattering of photons, now known as the Raman effect. 2 years later, in 1930, he received the Nobel Prize for his groundbreaking discovery, which marked the first Nobel Prize for India in the field of science. Our country has been celebrating the National Science Day since 1987 to create scientific awareness, to honor scientific achievements, and to spark conversation about the ongoing issues and to implement newer technologies for the betterment of our society. I'd like to mention one of C.V. Raman's great statements, and I quote, "Success can come to you by courageous devotion to the task lying in front of you." Every year, in his honor, we celebrate the National Science Day with a particular theme. This year, the theme is Future of STI, Impacts on Education, Skills and Work. With that in mind, I'd like to invite Madhura to please introduce our esteemed speaker for the day.
Good morning, everyone, and thank you so much, Vishakha, for that lovely introduction. So, we have a very accomplished personality among us. Welcome, Dr. Sanket Bapat, sir. So, I'll give a little introduction about sir. Dr. Sanket Bapat received his bachelor's degree in biotech and a master's degree in bioinformatics from University of Mumbai. He's worked as a part of a project fellow in Hafkin Institute of Training, Testing and Research at Parel in Mumbai, where he has worked on identifying the host target proteins involved in swine flu. He later completed his PhD from the premier CSIR National Chemical Laboratory and the Biotechnology and Bioinformatics Institute, Pune. The focus of the PhD work was to have a complete study of protein-protein interactions in cancer using machine learning techniques and developed a robust classification and regression model. Sir has also published several research papers in peer-reviewed journals and has written a book chapter to his credit. He has received many accolades at international conferences for his papers. Apart from his research, he has a strong background in academic and institutional teaching experience. His research interests include machine learning, structural bioinformatics, molecular dynamics, proteomics, chemoinformatics, and genomic studies. He is currently a visiting faculty at the Department of Bioanalytical Sciences at Ramanarayana Rudya Autonomous College. He also is a reviewer for the ongoing project Manav, a Human Atlas initiative, which is organized by DBT and Persistent Labs Pune. He also manages a YouTube channel of his name on which he explains the working of basic bioinformatics tools. So, sir, I can really say that me and my classmates have found those videos very helpful. Thank you so much.
Thank you.
We are very grateful to have you with us, sir, and I would now request you to address the participants.
Yes, first of all, welcome everyone, and wishing all of you a happy Science Day. And I would like to thank Purvi ma'am for inviting me over here to give a talk on this day. And also I would like to welcome and thank all the professors and also the students who have joined here to listen to my talk. Hopefully I will bring justice to whatever the topic is. And actually yesterday Purvi ma'am told me that there's going to be a lecture for, I have to give a lecture of 1 hour, okay, regarding this topic. So, you know, it generally is that whenever an industry person or a researcher comes, it's like always a talk. But whenever a professor comes, it's always a lecture. Okay, so I would like to tell all the students out there, it's not a lecture, it's a talk. Okay, so enjoy the talk. And my topic would be on something called as Bioinformatics, a road less traveled. The reason for choosing this title, A Road Less Traveled, is probably you will get to know as we go on through the thing. And I want to give all of you a different perspective on my subject. Okay, that is bioinformatics. All of you obviously must be knowing about bioinformatics because we have graduate, postgraduate students and also PhD students out there. But I would want that all of us to give you a different perspective about what this subject is, and I have balanced the entire PPT in such a way so that even the undergraduates who are new to this field can understand, and even the super specialized people, okay, like Sir who's there, even he can enjoy the topic. All right, so if I start this topic and I do not mention Margaret Oakley Dayhoff, it would be a crime. So she was an American physicist, and she was also the pioneer in the field of bioinformatics. She was working with Robert Ledley in the Ledley lab, and what both of them did was they were the ones who actually predicted the first sequence for the amino acids which are there in the proteins. All right, so they had something called as an IBM 7090 system, okay, which was like a huge system. I think in the one which you can actually see, it looks something like a typewriter. All right, and the language which was used that time was Fortran language. So, so on the Fortran language, they could predict what the next sequences were. To give you exactly how it would have been, if you all have seen the movie of Alan Turing by Benedict Cumberbatch. All right, in that movie they could decode the Enigma code because it could predict what exactly it could be. So it was something of that sort. And in manual hours, if they were to analyze what the sequence would be, it would have taken months and months. But using her coding experience in Fortran, she could decode what the sequence was in a short time. And what they both did was, in 1962, the first ever paper on application of computer program in biology was published with the title called as ComProtein. Okay, so this was the first ever paper in 1962, which was by Margaret and Robert, where they actually presented all their work of how this protein amino acid sequence could actually be predicted. And this work laid to a foundation for a new field altogether. So people got to know that we could implement informatics in biology also. Right, and based on this, they started working more on this system. And afterwards, in 1965, they came up with. So initially, they just worked on one sort of protein, and not also on the amino acid sequence. Then finally, they got to know that they could actually analyze the entire structure for the protein. And that is when they came up with the atlas for proteins structure and sequence, which contained a repository of all the proteins which are available. Okay, and this later, as you all know, in 1995, paved the way for RCSBPDB, or Protein Data Bank. Okay, I think all of you out there must be already well aware with the Protein Data Bank. What is that? But the origin of, so I can say the origin story of Protein Data Bank was actually the Atlas of Protein Sequence and Structures, which was started way back by Margaret Dayhoff. And slowly and slowly, the world started to acknowledge that application of computation, the application of computers in biology could be of a great help. And finally, by Pauline Hedwig and Ben Hesper in 1970, they came up with the term, or they coined the term, which you and me today know it as bioinformatics. So the journey started way back, okay, and it took around many, like 20-30 years for the field to be accepted by everyone. And then it was like, okay, there is something where we could use computers as an application for biological analysis. So what exactly was bioinformatics is nothing but it is a field that develops methods and software tools for understanding your biological data. Okay, so you can use various softwares, you can use various tools, okay, in order to understand how the DNA, how the protein, how the every other system which is there, it actually works, how the entire biomolecules they work. So that is bioinformatics. Then what is computational biology? Can you all tell me, okay, what is computational biology? Are both of them same or both of them different? So the one who's managing the chat can tell me. Yeah,
I think
yes,
both
sir.
of them are different, and
Okay,
bioinformatics is more to do with developing tools to actually analyze the data, and computational biology is basically, I think, trying to come up with new ways to analyze data.
okay, all right. Any other answer? I think best instead of putting the chat yourself in answer, that will be much quicker. Okay. So computational biology. So now, if you go on Google and if you type what is the difference between bioinformatics and computational biology, almost all of them will say both of them are exactly similar. But there's a very, very thin line between what is the difference between a computational biology and bioinformatics. So some suggest that a computational biology is nothing but the field where there is a use of softwares and tools for biological analysis. Now look at the difference between both of them. In bioinformatics, you are developing the methods and softwares for analyzing the biological data. And in computational biology, what you're actually trying to do is you're using the softwares and tools for biological data. That means everything what you all are doing, okay, you're using BLAST, you're using clustral omega, okay, you're using different sorts of databases. So that's something which is part of computational biology. But when you call yourself as a bioinformatician, then you have to develop those softwares, you have to develop those algorithms, you have to develop those methods. All right. So when you are again, it's a very thin line between both of them. Some of them suggest both is exactly the same, but after reading many papers, I found few of them which said that in one it is developing and another it is using of the softwares and the tools. So try and understand what I'm going to, what you know, okay, is all part of computational biology. But when you call yourself a bioinformatician, there you should be able to use other tools. All right. So yeah. Yeah. So I want to ask you all, what is the population of uh, the world? Approx.
7.8 billion people.
Very good. So there is approximately 7.8 billion people which is there around. And I'm calling 7.8 billion people or 7 billion people. All right. For you and me as a bioinformatician, okay, I will call this as a I will call this 7 billion. For you and me, it will be called as a data. Okay, so each and every each and every sample which is there, for us it's like a data. Okay, and yeah, so 7 billion population of the world. All right, and for us every 7 billion population is nothing but a data. And the entire bioinformatics deals with, okay, the entire bioinformatics deals with handling this huge data. All right. So and when we come to the concept of bioinformatics, in here we will have something like your DNA data, okay, or your blood samples data. All right. So or it could be something like a chromosomal data. So all of this, okay, is can you see the slides being changed?
Yeah, now it has changed. It's
Yeah,
showing the
okay,
PCR
okay.
machine. Yeah.
Yeah, okay, okay, okay. So it's taking, I think, some time to
Load it.
change it, load it. Yeah. All right. Okay. Anyways, so when you're talking about bioinformatics, we are talking about all this sort of data. We're talking about the DNA data. We're talking about the gene data. And where does this play a role when it comes to you experimental students out there? Is you all generate a huge amount of data. Okay, there's constantly a working the labs, constantly data is being generated. Okay, so your role comes is when you can compile all of that information and create a database. Okay, all of you must be knowing about NIRH, which is there in Lower Parel. So they generate a huge amount of NMR data, HPLC data, GC data, and what they do is they give it to the bioinformatics department and they compile them to form or create a database. All right. So because see, the data is generated from the experimental people. Now, how to compile the data and analyze the data is where we come into picture, which are the bioinformaticians. So why do bioinformatics? Two reasons. One is there's a huge amount of data which was being generated. All right. And there was a need to bring an order to this huge amount of data. So that's why bioinformatics came into being. And second is something called as analysis of this large scale of data. These were the two major reasons why bioinformatics was brought in. All right. So when I'm saying analysis of large scale data, I'm not saying like 50, 60 samples. If you do something like an experiment, you want to study the expression studies of a gene, okay, experimentally you can do about 100, 200. Okay, but if you want to do expression studies when it uses a computational methods, you can do about 1 lakh, 2 lakh of them, because all of this you have to run basically on the system itself. Okay, so what should I say bioinformatics is? Bioinformatics simulates the environment of the experimental conditions. Okay, please try and understand, I've used the word simulate and not stimulate. Okay, this is where most of the students get confused. All right, stimulation is something else. Simulation is where I am creating an environment which mimics your real experimental conditions. That is all bioinformatics is all about. And because of such huge and once the advancement of now, if you go back to the age of what Margaret Dayhoff, that time the computers were not even invented. But nowadays, in the past 20 years, 22 years, everyone has a system at home. And because everyone has a system at home, now everyone is generating the data. All right. So that is why the use and the need for bioinformatics has actually increased. And if you look at the global bioinformatics market, okay, way back in 2018, it was about 7.3. And now they are saying by 2025, it can go to about $13 billion. Okay, so that is the demand for this field. And there are lot many investors who are actually investing in this field of bioinformatics. In what field, if you ask me, as I go ahead in my slide, I'll tell you what all fields is the investment being going on. So bioinformatics is the future, and there is a huge amount of money which is being pumped in for analysis of the bioinformatics. And if you ask, as the thing it is, it's the global bioinformatics market. So this was a recent report of global bioinformatics market report of 2020, which had come. This is of last year. 2021 has just been released right now. And from there, I have taken this data, and they say that in India also there has been a huge investment of money which has taken place. So a lot of foreign companies are actually investing in India to carry out their analysis. Okay, see, one best thing about India is there is a huge amount of labor, and that's why the analytical part is what can be done to a greater extent when it comes to India. So there are a lot of companies in Mumbai. There are a lot of companies. There are much more lot of companies in Pune, in Hyderabad, in Bangalore, where the demand for bioinformatics is high. So if you ask me what is the range which the bioinformatics actually covers, one is from cellular mechanism, second is the metabolomics. All right, so one, it talks about the entire cell. So if I'm talking about the cell membrane and what all proteins which are present in the cell. If I go much deeper inside, it talks about the metabolomics, that is the metabolic pathways, the study of these metabolic pathways, the study of the signaling pathways. All right, then more deep, if I go, it talks about proteomics. So when I'm talking about signaling pathway, it is nothing but two proteins which are interacting with each other. Okay, so when I'm talking, so from metabolomics, the bioinformatics can also go much deeper inside to study the proteomics. And finally, you can also analyze the sequence of each and every of these proteins. So the sequence can be of proteins, the sequence can be of the genes, it can be of the DNA also. So just look at the range of the analysis which bioinformatics can do, right from the cell to a very small thing to the most smallest thing, that is your sequence analysis. It covers everything. All right. So when I'm talking about cell, when I'm talking about a sequence, and if I do not mention about the omics approach, then it will be wrong. Okay, so omics approach involves, as you can see, okay, you can have the genomics approach, you can have the transcriptomics approach, the genomics, okay, the proteomics, the interactomics, all right, the reactomics, the metabolomics, and the phenomics. Okay, so these are the various studies. Okay, so genomic study is the study of your genome. Proteomics study is the study of your how the different transcriptions takes place. Epigenomics is the study of the epigenetics. Then you have proteomics, which is study of your entire proteome. So it's not about basic protein, it's about how the entire large scale of protein take place. Interactomic study is the study of your protein and protein interaction. So how the two proteins interact with each other and what happens. Reactomics here, it actually suggests about your reactions. Okay, so between your protein and the metabolite, what various reactions take place is described in your reactomics. In metabolomics, it gives you the information about the different metabolic pathways. Okay, and phenomics is nothing but the phenotypic studies. At the end, when everything happens, what is there. There's also microbiome studies which can also be done using the omics approach, which is the entire information about the microorganisms which is present. And I don't know if you can see the graph which is there on the right hand side, but if you cannot, I'll just probably you can see the graph, but you cannot see what is written below. All right, the one which is in the blue, the top, the bottommost region, you can see the blue, the dark blue bar. That is suggests is the bioinformatics market from 2016 to 23, and the US, the number of millions of dollars which has invested. So you can see the highest number of dollars, highest amount of money has been invested in the genomics area. All right. After genomics, it is the drug design, which is nothing but your pharmaceutical companies. All right. So genome has a huge market. All right. So when I'm talking about genome has a huge market, then the next question comes is, what exactly is the market are we talking about? So if I compare between your genomics and your proteomics, the genomics was actually started when we began with the Human Genome Project way back in 2003, when the entire genome of the humans was actually coded. And there was a big amount of data which was generated. But that time, the computational power, what we had that time, was less as compared to now. Right now, if you want to do Human Genome Project, it will be that time it took about 3 to 4 years, and right now it will be done at a much faster pace than what it was done before. All right, so there is something called as 3 generations of next generation sequencing. All right, so so the first generation of your sequencing is basically what involves your electrophoresis. It's something called as your 2D page. So whatever you all are doing, okay, that is your first generation. The second generation is something called as the next generation sequencing. So in short, if I want to explain you what exactly is next generation sequencing, if I have the entire genome of any organism, okay, I can understand, I can annotate its genes, I can annotate the functions of all the genes in a couple of minutes. Okay, that is called as next generation sequencing. I can analyze how the genes are placed. I can analyze what the sequence is going to be. All right, that is all part of your next generation sequencing. All right, and the third generation sequencing is development of much better tools which can do the same sort of analysis in a couple of seconds. All right, and there are systems which are present which can do that. All right, in a very short period of time. So so when I was talking about there is a big revenue when it comes to genomics, there's actually a big revenue when it comes to this particular thing, that is your next generation sequencing. Again, probably if my image is small, I'll just tell you, this is a screenshot which I've taken of Naukri.com. And if you just type next generation sequencing jobs, I received somewhere close to 1,000, is it? No, it's about 10,000 jobs which are available right now all over India for next generation sequencing only. And you can do that. You can go to Naukri.com and type NGS. So the whole point of showing you this image was people are like, "Ha, images batate hai, itna paisa dalte hai, ye hai," but where is the reality? So if you go to the Naukri, if you go to any of these sites, yes, there's a huge demand for NGS people. All right, and as I said, there are a lot of institutes, right, from big companies like Serum or Syngene. Okay, they are all investing a lot, or Schrodinger, they are investing a lot on NGS, and there are very small startups also which have begun, which are moving on in the genomics section. So genomics is basically a future, all right, when it comes to bioinformatics. But proteomics is not behind too. My entire specialization was done in the proteomics part. So like in Human Genome Project was started way back. For me, I mean, for basically proteomics, we also have the Human Proteome Project, which is actually still going on. A part of it was actually released even in Nature way back, where scientists from all over the world are trying to analyze what is the different protein of the human body. And when I'm talking about the different proteins of the human body, I'm actually talking about the protein and the protein interaction. All right. So if I'm talking about the two proteins interacting with each other, what is very important here is this yellow region. Can you see this yellow region? This yellow region is called as the interface region. Okay. The interface region, or the active site, is where the two proteins actually interact with each other. All right. And more, if you go into details of proteomics, it is something which is called as protein folding. All right, there is immense studies which are going on protein folding, and as we go ahead, I'll also tell you how one of a major company has actually worked on the protein folding part. And why is it important to? But why is it important to study protein folding? Is the protein is basically the functional aspect of the body. So all your functions are actually initiated by your protein, and how your structure of the protein determines how the function is going to be. All right. So that's why understanding the protein-protein interaction and understanding how the protein is going to fold plays a very important role. So genomics and proteomics. All right. That is the study of the genome and the study of the protein. All right. Where one of the biggest applications, I will say, of bioinformatics. All right. If I'm talking about the sequence, which I had initially, I cannot, I have to talk about the phylogenetic analysis. All right. Phylogenetic analysis is where you can analyze various sequences with each other in order to understand how many sequences are related to each other, how many sequences come under one family. That is completely based on sequence alignment. Now, I know, I mean, I hope that all of you, I mean, as ma'am said, all of you are from postgraduate, right? Or postgraduate and PhD students. So phylogenetics is not something new to you. All right, this is the work which so I'll just tell you what this work was. This is what we had done about diabetes complexes. We had taken all the diabetes complexes and we had done a phylogenetic analysis of them, and we wanted to see how many of them are related to each other. So this was more of at a sequence level and a functional level. What is the relationship between the different classes of the diabetes complexes? And it was published in 2016, this paper, where we have analyzed the entire, where we analyzed all the complexes which are present of diabetes, which are present on the time. All right. Similarly, studies we have done for the COVID strains. We have got some really interesting results, and the paper has been accepted. And the reason I'm not showing the image is because it has yet to be published. But once it gets published, definitely. You can have view, where we did phylogenetic analysis of all the strains which are there of the COVID, and we observed some interesting results which were there between all of them. So there were mutations, and some of them had mutations in the functional regions also, and that could play a part. That might play a part actually when it comes to development of any drug. But now we are anyway moved to the vaccine part. So yeah, this was done last year. So you know, it takes time sometimes for publications. All right. The third part is, as I talked about drug, so I need to talk about drug discovery and development. Okay, I think Purvi ma'am, who has attended my molecular docking lecture, and also some of you students who might have attended that session at RUIA, must be aware of this slide. But I've kept it for those who are new to this. So the first one is where, if this is the process of identifying or discovering a new drug. How to do it? You first identify the disease. Then you isolate the protein which is involved in the disease. Third is to find an effective drug which is present for that disease. Next is you carry out preclinical testing. So after you have found out the drug, you do in vivo, in vitro tests, okay, or on cell lines, basically. That's a preclinical testing. After you have done that, then you increase the size of that. So because all of that has been done on a small level, the testing has been done on a small level. What if I increase the quantity? Will its efficacy or will its efficiency or the affinity of the drug towards the protein decrease? All right. So that's where in the companies you carry out the scale up and the formulation. After it's successful at that level, then you file for an investigational new drug. And after it's successful, after it has passed all the norms, then you go for human clinical trials. And after the human clinical trials are done, then you file for an NDA or a new drug application. And after that, after all of this process done, then finally the FDA approved drug comes out in the market. All right. So this is the entire process of of any drug, okay, which you have at your home, which you have it in your bag. This is the entire this is the entire process for it to come out in the market. Now, quickly, I want you guys to look at the number of years. Okay. Isolating the protein, effective drug, 5 + 5 is 10 years. Preclinical testing, 3 years. So 10 + 3 is 13. 13 + 10 is 23. And 23 + 3 is 26. So this is at a higher number. So it takes about 26 years or somewhere close to 15 years nowadays for any drug to actually come out in the market. Yes, that much time it's actually taking for any of your drugs, which is done by Ranbaxy, by Glaxo, by Cipla. For any of these drugs to come out, that's the time which has been required. So something like in pandemic, for any drug to come out, the reason why they were not so successful is because the number of human clinical trials which were done were very less. All right. If you see, it takes about 10 years for human clinical trial, because after the drug has been administered, the one has to see how much time does it take for it to be effective against that. All right. So that's why it takes a really lot of time for any drug to come out in the market. And imagine at any point of this it fails, okay, there is a huge loss for the company. Now, this slide, I always like this slide is because for all the students out there, okay, there are multiple job opportunities. Okay, so if you go from identifying the disease to isolating the protein to preclinical testing, that whole section is a part of something called as bioinformatics. What I'll be telling you, okay, how bioinformatics is involved in that part is what I'll be telling in the further slides. So that's all the bioinformatics section. So if you learn bioinformatics and you go into any pharmaceutical company, you could do the previous part. So that is in the R&D department of this. All right. Finding effective drug, this part is more of the biotechnology or the microbiology part, which you guys are already doing and you are already expert at. Okay. Filing of IND and NDA. Now these two sections, okay. Oh, sorry. Before that, scale up and formulation. This is more of the analytical part. All right. So the students who have done bioanalyticals, okay, or who know GC or who know what is HPLC, all right, to analyze the drug, to find the distribution of the drug. So if like something like a drug administration, okay. So if I'm to administer the drug, should I make it topical? Should I make that drug as an oral way, or should be an intravenous? And if it's a tablet, then what should be the distribution of the drug in that circle? Okay, so it's the circle, right? The tablet. So the drug should be equally distributed. All of that is a part of your analytical part. So that's a bioanalytical part what we are talking about, and a pharmaceutical part. So that is another job opportunity. Then you have IND and NDA. Now this thing comes under something which is called as regulatory affairs. All right, so there is a course for regulatory affairs for a few months which you can do, and after doing that course, you can join into any CRO, that is a clinical research organization company. One of the biggest. Does anyone know the name of one of the topmost CRO in the world? Now it has become second, but that company is there in India. That's why I want to tell you. I want to ask you, do you know the name of any CRO company? That is clinical research organization company.
I'm not exactly sure, sir, but let us throw this question to our participants.
Yeah.
Please unmute yourself and speak.
Okay. All right. So no one wrote in the chat. Okay. So students, one of the biggest CRO. Okay. For the past 10 years, it was #1. Just 2 years back, it came down to #2. But I think it's going back to #1. They're working on it. That is called as IQVIA, or the Quintiles. Okay, that's one of the biggest clinical research organization company where they actually carry out regulatory affairs, and they do this IND and the NDA regulation. All right, and if you ask me where exactly is Quintiles or IQVIA, that is there in Thane. All right, such a big company. Okay, which it's an MNC. All right, and they pay also very well. It's present in Thane itself. It's present in Mumbai. It's present in Thane. So yeah. So this is another scope for you guys. Human clinical trials. Don't think that you will do clinical research and then you will do human clinical trials. No, that is only for the doctors. So in just this one slide, you have so many opportunities which is available. All right. I know there's already a topic of what are the job opportunities after bachelor's or master's. A lecture has already been taken for you all, I think. Correct, Purvi ma'am? If I'm correct? Yeah.
Like usually in our placement cell,
Yes.
such things are regularly discussed, with the students.
Yes. Yeah. So no, I think.
Yeah. In fact, even IQVIA, what you mentioned, we have some of our alumni who are working there.
Okay.
So,
Okay. That's great. Yeah. So students, you should know where your seniors are working. That's the #1 thing when it comes to finding a job. All right. So you should know where your seniors are working, and you should be searching more. Okay. So yeah. So understood what exactly is drug discovery and development. All right. So just want to tell you that what is the cost proportional to degree of difficulty. The cost of Mangalyaan was about $71 million. The cost of Mars rover was $3 billion, which was sent by US. and the cost of developing one drug is about $3 billion. All right, that's how costly it is for any drug to come out in the market. That's why if you compare it to the generic drugs, these drugs are also very costly. All right, so it's a very that's why if you mess up with any of them, the company could have faced a huge loss. All right. So as I talked about the initial section, which was identifying the disease, isolating the protein, and identifying the drug. All right. Now, as I told you, that bioinformatics plays a very important role. So yeah. So in that section, okay, there's something which is called as a personalized medicine. All right. So personalized medicine is where you develop a medicine which is specific for you. So what happens is if there's an individual suffering from breast cancer, you do the molecular profiling for it. And then after you do the molecular profiling, which means you look at its genomic profile, you look at its proteomic profile, you look at its metabolomic profile, and you look at its transcriptomics profile. And after that has done is you identify a drug based on what is there. You identify what the drug is and which can be specific for that individual. And then you go for a treatment. All right. Because generally what we do is we make drugs which is for the and that, like something like crocin, okay, or paracetamol. So paracetamol, everyone and anyone can take it, but there are people who cannot, for whom paracetamol cannot actually function or work. So and it's our duty to treat them, as a researcher, to even make them healthy. So that is where the concept of personalized medicine has come, and there's a huge work which has been going on in personalized medicine. One interesting fact I'll tell you is that Ayurveda actually has been following personalized medicine for a very long time. The problem is we are so much we are so much busy looking at the West that we forget that what all great things we have here. If you go to an Ayurvedic person, he asks you all the things. Okay, are you suffering from anything? So he asks you all the medical conditions, what you have, if you're suffering any issues which you'll be having, and based on that, I mean, nowadays I don't know, but initially they used to prepare that medicine and give that powder to you. So Indians have been following personalized medicine for a very long time. So this is another great research area which people are working on currently. That is the personalized medicine, and yes, bioinformatics plays a very important role because it involves the genomics and the proteomics areas. All right. So yeah, another section is your chemoinformatics section. So this is all we are talking. It is a part of your drug discovery and development. So these are the sub specializations which are part of the drug discovery. All right. Chemoinformatics is application of IT in chemistry. I think. As far as what ma'am told me, there are some students of chemistry background, and there are also some faculties from chemistry background. I think they might have joined. Yeah. So I think this is something which will interest them, and if not them, it's also even for the biologists also out there. It's when you're talking about a drug is nothing but a small molecule. It's a small molecule. Okay, it's a compound. Now, what all can you do? Okay. Can you make analogs of that compound? Can you make derivatives of that compound so that it basically binds to the protein, to our target protein? That is all what is part of chemoinformatics. All right, to develop new drugs, to develop better derivatives of drugs, basically. All right, and in order to understand or know chemoinformatics, you need to know chemistry, you need to know statistics, you need to know informatics, and also you need to know mathematics. Statistics and mathematics is because you need to understand how to develop an algorithm, or you need to develop a software in which we can analyze the protein structure. All right, because knowledge of chemistry obviously is important. Okay, you should know what is a scaffold. You should know what is the functional groups which are added to it, and what functional groups you change. How is the bioactivity of the molecule going to change? Okay, so that's why chemoinformatics is another big thing. My PhD also involved some section of my PhD also involved chemoinformatics, because my guide was is actually a chemoinformatician. So I had to learn chemistry because of that. And one more thing before I move ahead. Two things which all of you are scared of, that is your mathematics and statistics. All right. The reason I know why all of you have taken biotechnology or life sciences, because you do not like mathematics. But surprise, surprise, mathematics and statistics is a very important part of bioinformatics and also of biology. You have something called as biostatistics and all. But when it comes to bioinformatics, you need to know this. Also try and understand, we have already moved way much ahead, but also try to understand one thing. The pioneer of bioinformatics was actually a chemist. Okay, so she was the one who actually started with this. Okay, she also had a good mathematical background. All right, that's why she could do the coding on the Fortran language. All right, so the pioneer was actually someone who was a chemist and a mathematician, and afterwards the rest of the ones whom you can see, they were all physicists. Okay, so so they were the ones who actually came up with this. So yeah, chemoinformatics, another great thing when it comes to the research part. All right. So when I'm talking about drug discovery, I talked about personalized medicine. I talked about chemoinformatics, and one section in that. Okay, I told you the initial section is all is all bioinformatics. Another section is what is docking. Obviously, all of you must have heard of molecular docking. So molecular docking is basically where I take the drug and I try to inhibit it with the protein. All right. So if you can see in this, this is the drug, what you can see, and around this is the protein, and basically this is the pocket region of the protein. All right. This is one of the work where we had tried to do docking of curcumin, okay, against the rest of the compounds which is there, and this was the interacting amino acids. All right, so this is another paper where we tried to do docking of chromone derivatives with the target protein. So this is where we had introduced both chemoinformatics as well as molecular, the molecular modeling part. So in this, what we had done, the chemoinformatics part, what we had done was we had synthesized various derivatives of the chromone compounds. We had also found out what its bioactivity was, and then based on its bioactivity, we had docked these compounds with our CSRC kinase. So CSRC kinase is a protein target for cancer, and chromone derivatives they have found out. Okay, the chromone derivatives they have found out, which is good against the CSRC kinase. And similar to this work, okay, yeah, this was the next thing was another slide. Similar to this work, we had done on tuberculosis also. So the initial thing was on SRC kinase. The other one what we had done was we had taken the same chromone derivatives, which were these, and we had docked them against tuberculosis also. Okay, because we found out that, but yeah, the difference between both of them is they are different. This is 1,2,3-triazole embedded chromone. They were different chromone derivatives. All right, so docking is one part. So after you have done the docking of the protein, the docking of the drug to the protein, all right, the next thing is to see whether both of this docking takes place correct or no. And one thing for those who don't know what docking is, it's based on this docking. If you get very good docking results, that is if your drug and the compound, the drug and the protein, they bind to each other very good, okay, then only in ahead it's very good for development of any drug. Okay, so that's why. So this docking plays a very, very important role. After we have done with docking, there is another thing which is called as a molecular dynamics. All right, so molecular dynamics, it actually simulates how your drug or your compound is going to go and bind to the protein. All right, so there is this video here. Yeah, can you see the video, everyone?
Yes, yes.
Yeah, okay. So what you see here is that yellow color thing, that is your compound, okay, and the blue colored thing is your protein. All right, and if you can see, the compound is going and docking itself to the protein. So when you actually do normal docking, it's done in a vacuum. When I say it's done in a vacuum, is you have a compound, you have a protein, you dock both of them together, that's it. Okay, and then you get the final result of what the pose is. When I'm talking about molecular dynamics, it's done in a liquid medium. The whites, that is basically in water. So you take the protein, I mean, this is all simulation. You don't physically take it. You take the protein, you put it into the water molecules. Okay, water molecules, because the maximum percentage which is present in our body is composed of water. So you take the water molecules, you take the protein, drop it into the water molecule, and then you try to dock the ligand with the protein. And and what happens is, because it's covered around with water molecules, okay, the water molecules can cause a steric hindrance to the protein itself, to the protein as well as to the ligand, okay, and both of them binding to each other. So molecular dynamics actually tells you that if it's successful in docking, will it actually be successful in reality also? Okay, because now in this case you can see it's been the ligand for a very long time. I'll just repeat it again. The ligand for a very long time has been settled in the pocket region. It has not moved anywhere. That means the docking here, what you have done in the previous step, is wonderful. It's great. Okay. It sometimes does happen. It had happened with us also many times, that we had done docking, we had got good results, but after doing molecular dynamics, the ligand had actually settled inside, but then it came out of the protein also. All right, and that is that is completely wrong. Okay, so that we cannot go ahead with that ligand for docking, or that compound for docking. So we selected some other compound, some other ligand. Okay, so molecular dynamics actually gives you the real life, gives you the actual virtual simulation of how the interaction between the compound and the protein is going to be. The other green thing, what you see, is another compound which has been already docked to it. Okay, and the yellow one is what we have actually docked. We have actually performed the docking with. All right, and the things what you see, the red color dots here and there, those are nothing but the water molecules. See, it's a simulation. So because it's a simulation, all the molecules are moving, all the atoms are also actually moving. So that is what it shows. Okay, and can you see the graph? Okay, the graph is pretty much stable. So graph also indicates that after a particular time, it is very much stable, and whatever we have done, this docking of the compound with the protein is wonderful, and we can take this compound and take it ahead for the clinical trials. All right. So this is what is molecular docking and molecular dynamics, and both are a part of something called as molecular modeling. So both of this play a very, very, very important role in your drug discovery and development. So you go to any pharma company, and if you are given an R&D when it comes to bioinformatics, this is where you guys are placed, and this is what your role is: to isolate the protein, to identify the compound, okay, then to do the docking and to perform molecular dynamics, and to see are you getting good results or not. All right, so yeah, so how does basically a drug move is you have absorption, you have distribution, you have metabolism, and then you have excretion. So that is your ADME properties. All right. So when I'm talking about ADME properties, I actually mean, so what is this? It's a quantitative study which describes how the movement of the drug takes place. And by doing that, what you want to do is you want to find what is the intensity of the effect, what is the concentration of the drug, and what is the site of the action. Okay. So it's a quantitative study. The movement of the drug is tracked in that. What is the intensity of the effect, like side effects or something like that? How much concentration of the drug should be imposed, and what is the site? Okay. So bioinformatics helps you to predict all of this. And how you can predict this is using pharmacokinetics. All right. So pharmacokinetics is a quantitative study of drug movement in and throughout the body. The intensity is related to the concentration of the drug at the site of the action, which depends on the pharmacokinetic properties. So pharmacokinetics is another one of the big application when it comes to drug discovery and development. All right. Yeah. And pharmacology students also work on pharmacokinetics, but even you and me as a life science student or as a bioinformatician can also work on it. All right. When we have talked about drug discovery and what is the applications of bioinformatics basically in the drug discovery, but there's one thing which I had mentioned initially, that was large scale of data. And when I'm talking about large scale of data, I'm talking about big data. All right. So I don't know if you have heard of this word called as big data analysis. Have you heard of this word called as big data analysis? Yes.
So I've heard of it, but not sure of the definitions. If any of the participants know, they can speak up.
Anyone? Do you know what is big data analysis?
Sir, may I try?
Yeah, please.
Yeah, so I think big data is when an extremely large amount of data is generated, which is not possible to analyze using any conventional methods.
Yeah, correct. That's the definition of it. Okay, so big data nowadays, and when I'm talking about big data analysis, it does not. It could be in anything. Okay, it could be a big data analysis of your molecular dynamics. It could be a big data analysis of your genome. So genomics, all right, or your NGS is a part of your big data analysis. All right. So and what happens in your big data is you want to finally do management of this data. Okay, so you have a big data, you want to manage this data. And how do you do management of this data is by carrying out this method which is called as data mining. All right, I don't know if you've heard of data mining, but data mining is another huge application in bioinformatics. Okay, it's a part of your data analytics part. I don't know if you've again heard of data analytics, but data analytics and data mining is where I mean they're not exactly the same. Data analytics is where you're analyzing the data, and data mining is where you're mining the data from this huge big data. What is there? So one I want to show you. Yeah. So the difference between data searching and data mining is in data searching, what like your Google search. So Google search is basically called as your data search. So if I type the story of in Google, all right, it's going to give me all the hits for the story of. So that is called as Google search. Okay, so it's giving me what all is there. All right, but when I'm doing data mining, it's trying to identify something, a particular specified type of string or something very unique in that. Okay, so if I type the story of and then I put a star in front of it, it's going to give me the hits for the story of stuff, the story of life, the story of philosophy. Okay, so it's not so. This is the difference between what is called as data searching and data mining. Okay, big difference between both of them. All right, and so data mining is another big thing because of this huge amount of data which is being generated. If you want to identify all the genes which are present in cancer, if you want to identify the drugs which are there in cancer, for that from this huge literature, that is where data mining plays a very critical role. All right. Sorry, I'll give you one example where we had done data mining. We wanted to analyze what are the various drugs which are present in marine organisms. So marine organisms, they secrete some sort of secondary metabolites which are effective inhibitors. So so this was a collab with National Institute of Oceanography, what we had done. So we took the names of all the marine organisms, the inhibitor, then we tried searching them using data mining in about 2 lakh research papers about what exactly are the inhibitors which they are present. We tried to associate those inhibitors with the the organisms, all right, and then we created a database with them, all right, and that database can be available for you. Okay, this is, if you see the last thing, is a chemoinformatics approach for building molecular networks and marine organisms. All right, this data, and the name of the database is called as I mean, it was decided by NIO. So the name of the database was called as MIMO. All right. The reason why it is called as MIMO, because that time finding Nemo. Okay, I think that's the name of the cartoon. Correct me if I'm wrong. Okay, which was it was very successful that time. So that's why they were like, let's name it as MIMO. That is marine inhibitors in marine organisms. Okay. Yeah, so so this is where one of the application what we had done for. So in a huge large scale data, we are training something fun. Okay, I think I started 11:08 according to my this thing. So I have still 5-6 minutes more, and I will complete it. Okay.
Yeah.
So the next application is basically in your protein modeling and protein and network generation. So in this, so I had talked about the marine organisms thing. So we had created a network, and how did we get to know that, which is the unique inhibitors? So this is where your pathway modeling and network generation plays a very important role. So what you do is you take your, so in this case, we had taken the list of all the inhibitors, we had taken the list of all the marine organisms, and we had created a network out of it. And in this, we found out very unique interaction of some drugs or some inhibitors, which we didn't know was present in some other organism. All right. So it could be something as simple as this, or it could be as complicated, something like this. This was the interaction of all the targets of malaria with the drugs, and what we wanted to do was we wanted to find out, in this case, all right, we wanted to find out what exactly are the biomarkers for the malaria using this interaction network. All right, now when I'm talking about pathway modeling and network generation, it's nothing but you are creating a network. Okay, and by creating the network, you're trying to analyze how the different proteins are interacting with each other. All right. In the introduction, you said that I had one book chapter was published. So one of the book chapters, which was published in Springer, was a part of pathway modeling. All right, where we had worked on non-small cell lung cancer and how the different proteins of non-small cell lung cancer are interacting. Now, all of this is a part of something called as systems biology. All right, so if you've not heard of systems biology tells us how the various pathways work. Okay, how the different proteins can be inhibiting each other. What is the expression of the different proteins or the different genes when they are interacting with each other. All of this is something which is part of what we call as your systems biology. All right. Yeah. So the next thing, another important part is what is immunoinformatics. So we moved from drug discovery to big data analysis, to protein interaction network. That is also part of your big data analysis. And next is your immunoinformatics. Immunoinformatics talks about the study of IT in the immunological studies. So you have your antigen, you have your corresponding antibodies, and you want to test the antigenicity. That could be done using bioinformatics studies. If you want to see if your antigen can induce any sort of immune response, you can check the immunogenicity again by you can predict it again by using bioinformatics tools. And if you want to predict the epitope, all right, with which the antigen is actually interacting, that could also be done using bioinformatics analysis. And the use for epitope prediction is for understanding the disease, is for host pathogen interaction, antimicrobial, and obviously your vaccine design. So it's not just the drug. The reason for showing you this slide is when we are talking about a drug discovery and development thing, it's all about developing a drug. And the one thing about drug is, all of you know, it's a part-time thing. It's a part thing. All right. Its effect does not last long. But that, when you look at the vaccine, once you give a vaccine, you don't need to, I mean, it protects you for a long time, like what for the COVID. So in COVID also, the vaccine design, what is being taken place, okay, this covaxin one, there were some immunoinformatics studies which were carried out by Serum, all right, for developing the vaccine, and mostly all of them do that for epitope prediction. But one thing I'll tell you in immunoinformatics study, when it comes to vaccine design, like in your drug discovery and development, your molecular docking and dynamics plays a very, very important role. It's only when it is successful there, then only it moves ahead. All right. In vaccine design, even if the epitope prediction is also correct or no, but finally the vaccine design is so tricky that you will have to do experimental studies in order to combine that. But this is also immunologists use immunoinformatics if they want to understand the epitope region. They want to also understand the paratope region if they like. Okay, so this is the immunoinformatics. And obviously, when I'm saying prediction, there are various algorithms which are used in prediction. Okay, and so there is something called as Markov chain. There's something called as Bayesian networks. There is something called as so there are various statistical methods in which you can identify them. And one of the way to identify them, one of the important part in immunoinformatics is this last application which I'm going to discuss right now. So tell me, what do you see here, all of you? Can anyone tell me what do you see here?
Owl and apple.
This is everyone. Owl and apple, very good. Now?
Muffin and dog.
Muffin and dog, great. Okay. Now, this is obviously all of you know this is chess, all right, and this is your game what you play. Okay, the like your PUBG or your Call of Duty or any other game what you're playing. What is common in all of this is this field which is called as artificial intelligence. All right, when you're playing a chess with the computer, what pieces you play based on how it is going to react to it. So that is all based on AI. So it has been programmed in such a way. When you're playing games here, okay, how you actually go about now, those who play games will know that when you're shooting someone or something of that sort. Now, I'm not talking about the multiplayer kind. No? I'm talking about your single player. All right. So sometimes when you're playing any game, your environment reacts to it. Suppose you shoot someone, then other people also start shooting at you, or something. The environment reacts some way, or it is. All right. When I was actually thinking of an example to give you this, I just realized that all the games relate to is shooting and a lot of violence. Okay, I could not think of any good example, non-violent game example where I could think of artificial intelligence. There might be any, but it was all thing. All right. Okay. So that's part of your machine learning here. Okay. Yeah. And what you see on the top is nothing part of your image processing. All right. So what Google's image search does it for you, it does something called as image processing. Now, you understood there's an owl and an apple. Now, you understood there is a chihuahua or a dog and a muffin. Okay. But how to train the machine, how to train the computer, how to train the algorithm to differentiate between both. All right. Because for them, both the patterns are exactly alike. All right, so that's where this advent of machine learning came into being. So the biggest application of when it comes to bioinformatics is AI or artificial intelligence. Okay. What you try to do in artificial intelligence or machine learning. Now, when I speak about artificial intelligence or machine learning, generally the thing what comes in your head is robots, okay, the robots taking over the world. Okay, that's not the case always. There's a lot in which when it comes to biological aspects, you have, you can use AI or machine learning. Okay. Now, what is the difference between AI and machine learning? AI is where machines and programs are given an ability to think and mimic. See, the biggest difference between you and a computer is logic. We can use logic to deduce problem. A computer cannot use logic to deduce the problem. But what if you start giving the computer a logical thinking, this cognitive ability? All right. So that's when, I said you're trying to make it more human, and that's what's actually happening. Okay. So AI is that. Machine learning is a part of your artificial intelligence. So machine learning is a part of AI, and what it does is it develops tools and softwares for carrying out this cognitive ability, for understanding the patterns, to identify or analyze the various things. Okay. Something like now, when you look at the biological terms or biological applications for that, many of the hospitals are trying to use artificial intelligence in their system. I'm not talking about robotics here again. I'm talking about analysis of MRI scans. Okay, analysis of CT scans. So there was one lecture which I had heard. So they were like, the number of people who are there to analyze the MRIs and CT scans, okay, they are very less, and the data which is being generated for MRI and CT scans on a daily basis is a lot. Okay. So that's why, I mean, when you do the MRI and CT, the results, I mean, you can see it in your screen or the results come quickly. Okay, but to analyze the problem, the doctors can do it sometimes, but to analyze the problem, sometimes it takes something like sonography results also. Right? So the doctor tells you at that time, but for a report to get generated, it takes a lot of time because the report is done manually. Okay, so an individual sits, looks at it, and then types and does it. Okay, so it takes a lot of time because of huge amount of data. What if you give everything to a machine? The machine will analyze the patterns which are there in the MRI. They will analyze the patterns which are there in the sonography, and it will give you the results there and there itself. Okay, and that is what is the future which is happening. There is an application of image processing and all of that. All right. Another application of image processing was done when one of the scientists from Singapore had come to give a talk at our end. He used machine learning in order to differentiate between patients which are schizophrenic and patients which are normal. So he went to the hospitals, took their data, and tried to find out the difference between the schizophrenic patients and the normal patients. Just give me 2 to 3 minutes more, ma'am, and I'm going to complete. I know I'm way ahead of time, but there's a lot of things I wanted to talk about.
Yeah, no problem.
So, in this field, yeah, sorry. Yeah. The another thing which is coming in, all right, is something what is called as can you see this? Yeah, it's called as deep learning. One minute. Is what is called as deep learning. I hope you can see the slide, right?
Yes.
Yeah, it lagged somewhere in the middle. I'm sorry. Yeah. So deep learning is now a part of machine learning. So deep learning it actually mimics your human brain. I don't know if you've heard of deep learning, but actually mimics your human brain. So how your neural networks works, right? You have axons, cytons. So similarly, in this also you have various centers which interact with each other. So can you still see the screen?
Yeah.
Yes, so we can.
Okay. All right. Okay. Sorry. So how your this thing works is what is shown in your deep learning. So deep learning nowadays is set to be much accurate and better as compared to machine learning. All right. My major PhD, my 50% of the PhD involved machine learning. That's why I'm very passionate about talking about this. All right. This is just one example which I'm going to share with you about my machine learning part is where we had tried to differentiate between cancer patients and non-cancer patients. So what we had done was we had taken certain properties of I mean, if you have some samples in front of you, it takes a lot of time for them to determine whether it is having any tumor which is benign or malignant, or whether the patient is having cancer or not. So we had developed a machine learning model in which it could differentiate between cancerous patients and non-cancerous patients. And one of the parameters for it was using the binding energy between the different cancer complexes. Okay, so this could help us differentiate could predict, okay, whether any patient is having cancer or no, and even whether any protein can be a cancer complex or no. So the graph what you see, actually this was the output what we had got, all right. And based on this output, we had deduced it. So if you can see, there's called as a parity plot, and you can see the hyperplane in the center, and there were points. So a lot of points are there clustered near the hyperplane. And in this way, and by the way, there are two sections to it. So there's one of them which are cancerous, and the points on the other side are the non-cancerous one. And because as they're so much close to the hyperplane, that's why the accuracy was, we got around close to 94% of accuracy for generating it. Okay, so this was application of machine learning what we had done. And one of the biggest application right now what has been done by Google actually is Google has found out something called the DeepMind artificial intelligence, which is able to predict the protein folding. You remember I told you why protein folding is important initially, and yeah, this is what. So it's a recent Nature paper where now Google also jumped into this thing of AI. I mean, it was already a part of AI. It has jumped into the field of protein folding and bioinformatics in order to analyze this for us. All right. So the question now comes is what exactly is the career when it comes to bioinformatics? So I've talked about the education, okay? When it comes to bioinformatics, that is right from your data management to your big data analysis, to your drug discovery development, to chemoinformatics, pharmacokinetics, machine learning, okay? So that was the education part, all right? Next part is what exactly is my career ahead in this field. So either you could go into research, or you could go into industry, or you could be part of teaching. Now, when you want to go into research, let me tell you, you have to do a PhD, and if you want, you can do a postdoc also. Okay, if you want to make your career as a bioinformatician in research, then a PhD is a must. Then you can land in a very good institute, or you can land in any education institute also, like your SIRs and MAMS which are there. All right. In industry, you could do a graduate, you can be a postgraduate, and you can join any industry. Now, when I'm talking about industry here, most of the industry deals with your data analytics. Okay, that can be anything, genome, proteome, anything. You remember what I told you initially? There's a lot of market in this thing. That's more of the industry part. Okay, so a lot of data analytics and big data work happens in the industry part. And last is obviously the teaching part. Okay, but what I suggest is if you combine teaching and research together, then it's actually great. Okay, because if you are just doing teaching, you are confined in that small space. But if you combine both of them, it's really fantastic. And the third part, which I want to say, so yes, education, there's career, and there is skill. Okay, also which is the theme of this year's Science Day. Okay, so what skill set you will be requiring if you want to be successful in bioinformatics is coding. All right, so if you are very, very good at coding, if you love coding, then this field is for you. If you do not like coding, if you sitting in front of the system for a very long time troubles you, then please do not be a part of this institute. And it's a very small last slide which I want to tell you guys for those who are interested in research, because even though I am a teacher, but I identify myself as a researcher because I'm more into research. if you want to be into research, okay, intelligence is the must, okay. But more important than that, so suppose consider this as what is the foundation, okay, for this. So intelligence is at top, which is very important. But more than that is your concepts. More than that is your hard work. And the most important foundation for your research should be your patience. All right. If you do not have that much intelligence, if your concepts are not that too much strong, it should be strong, but not that too much. Okay, but you are very hardworking, you are ready to learn new things, and you have tremendous patience, then believe me, guys, research is for you. Okay, you can ask any of your faculty here who has done a PhD, and also your PhD scholars who are doing PhD right now. Okay, they'll all agree to this thing that like patience and hard work is the key. All right. Concepts is there, intelligence. Hey, yeah, intelligence though. Hey, but it's even if you don't know things, you will get to learn new things. But these two are the important part. And to my last slide is this is the in 1916 there was this poem, "The Road Not Taken" by Robert Frost. If you ask any person in literature, he will tell you about this. So there is one line, the last line of the poem suggests two roads diverged in the wood, and I took the one less traveled by. And that has made all the difference. Okay, so the reason for choosing my topic as the bioinformatics, the road less traveled, okay, does not mean that very few people go there. Okay, it means that it's not because even if very few people go there, it's not because people don't like it or there's no scope in that field. It's very few people go there, or it's less there because people are not aware of that field, of how important that field. If you ask any any life science student, bioinformatics पूछा तो sequence alignment, phylogenetic analysis, molecular docking में खत्म हो जाता है. इतना ही तो है. क्या है बायोइन्फॉर्मेटिक्स में? ये तो है डेटाबेस में बैठ के काम करना है, और क्या है? But today I know I went way beyond the thing, but I wanted to educate all of you that bioinformatics is not just that. Okay, so that's why any life science student who actually, it's like like Titanic, right? You all are sitting in the ship, all you life science students. तुमको सामने वो iceberg दिखता है, bioinformatics का अच्छा sequence alignment है, bioinformatics बहुत easy है. You like चला ठीक है, will go into it, kuch nahi hoga. But then that is just the peak of the iceberg. What lies below is you have machine learning, drug discovery, pharmacokinetics. There's so much, and that's what happens when you actually dash into this field of bioinformatics. Your ship collapses. All right, that's because there is much more to this bioinformatics than what you all know, and this is what I wanted to share with you all, and that is why. The title, which I don't know, first of if you have seen, thoda sa tum log confused ho gaya, ye kya title choose kiya hai. But this was the reason for the title, is because I tried my level best in, I exceeded my time of, I took like 1.5 hours or something, or 1 or 20 minutes. But I wanted you to know what exactly is bioinformatics, and what better day than Science Day to make you know about my field. And yeah, one more thing I forgot in that that pyramid which I showed is there's one more thing, okay, that is passion. If you're not passionate about this field, forget bioinformatics, you're not passionate about any of the field, biotechnology, molecular biology, or chemistry, or microbiology, whatever you guys are doing, you cannot succeed in this field. Okay, you can never. This is a passion driven field, and पैसा आता है, पैसा बहुत बाद में आता है. All right, if you run after money in this field, you will reach nowhere. Okay, but if you like this field and you want to do something in this, then only there is great success. You look at all your teachers, you look at all the great scientists, they have they have something for this. All right, and if you don't have passion, you cannot succeed at all in this field. All right, so in this field of research, basically. So thank you very much. I'm extremely sorry I went beyond the time.
No problem.
Not many parts, and I tried doing it. And thank you very much, ma'am.
Yeah. Thank you very much, Dr. Bapat. I think it was very well. I think you took showed the journey of this bioinformatics. And very rightly you said that, students normally feel that, okay, they know how to search for a sequence or align it or do the phylogenetic alignment, create a phylogenetic tree, and docking, that's the thing. But I think you really gave a broad insight of the different avenues, which can be studied. And of course, like you rightly mentioned, the statistics, the machine learning, these are the core, I would say, the foundation of this field. So thank you very much once again. And I think the floor is now open for questions. So, I think the moderators can ask questions, or at least ask the audience to, if they have any questions, they can even raise their hand and unmute themselves and ask it. Yes. I think it was quite a clear talk. So I don't think there are questions at the moment.
Sir, I have a question.
Okay.
Yes.
Oh,
Yeah, Suvarna.
Suvarna has also raised a hand. Let her ask first.
Okay. Suvarna, you can unmute yourself.
Yes, thank you very much, sir. It was indeed very informative talk. My question is, when you talked about the molecular docking, meaning what is the percent of success when this molecular docking is to be confirmed by the wet lab experiment?
Okay, so you mean to say okay, so after you perform docking, there's something called the docking score which gets generated. Now there is nothing like there's nothing like a perfect docking score. Okay, now if you have performed docking, you get docking scores like -8, -9, -10, -4. But there is nothing. It's not like कि अगर -8 है तो अच्छा है, -4 है तो बुरा है. It is only after you perform molecular docking and then you do MD simulations and then you see if your protein and your ligand have been with each other. Then only you can say that it was a good and successful docking. And finally, afterwards, when you go for preclinical testing, there is when you do docking, you're just concerned with the protein as well as the ligand. But when you actually incorporate it into the body of, like if you do in vivo and in vitro, there are various other factors which actually, play a role. So there you get to know clear picture of how exactly your molecule or your compound is effective. But molecular docking, it's good for narrowing down to specific number of compounds.
Okay. Yeah. Thank you, sir.
We have a question from Anushka. Anushka, you can unmute yourself.
Yes, sir. So first of all, it was a wonderful talk. My question is that we know that bioinformatics and experimental biology should go hand in hand. So I was wondering if there are any avenues in which bioinformatics can work in isolation, like it won't require a wet lab backing to the results. Is there any avenue that it can work like that?
Okay, now you mean isolation in the sense understanding the petri dish or something you're trying to, I mean, I don't understand.
No, as in an avenue where it won't require a wet lab like backing to the results.
Oh, okay, that way. No. See, I will tell you one thing. Okay, even if bioinformatics is done, you have to do an experimental. So if you mean to say that there will be a time where only bioinformatics, like IT-based work, will work, and you don't need to at all do experimental, that would never happen. You need to always go for an experimental thing. The reason being, so what bioinformatics actually plays a role is, it tries to, it's a process of elimination. Okay, so you have large amount of data, you have big data, it eliminates the unwanted thing, and it gives you that specific compounds or that specific data what you require. Okay, and then so something which experimental people cannot do. You cannot analyze thousands of data. You cannot analyze 1 lakh, 2 lakh data. I mean, you can, but it's going to take a lot of time. So that's where bioinformaticians come. So after we analyze this 2 lakh, 3 lakh data, and then we narrow it down to 10 to 20, then we can give this 10 to 20 data to the experimental to do it. So actually bioinformaticians and experimental people go hand in hand, is because our process is to from a lot to give you something very small, finite, and with that data you can work ahead. Okay, so I think this is what your question was, that there is something where bioinformatics will take over completely and no experimental will perform. Aisa kabhi nahi hoga. Was that your question?
Yes, that
Did
was
I answer it correctly?
Yes.
Okay, yeah, thank you.
Thank you.
Yeah, okay.
Yeah, so I think I would also like to add to what Dr. Bapat already mentioned, that if you are skeptical about the wet lab, you don't want to be, then you can always be a team where you are focused only on the bioinformatics approach. And like he rightly mentioned, from thousands of molecules, you narrow down to few hundred or few numbers and take it to the, pass it on to the lab, where in R&D they will take it up and validate it, So that validation part can be done by the other department. It's not necessary that the bioinformaticians have to sit and do the wet lab. No.
Yeah.
Yeah.
But one good thing about you all knowing bioinformatics is you already know wet lab. So if you know bioinformatics, that's the reason why bioinformatics is there in the curriculum in the first place. Okay, which none of the wet lab students, like you all understand. तुमको लगता है ऐसे ही डाला है, then you put, फिर तुम लोग उसको कि हाँ ठीक है, marks इतना है, इतना score कर लेंगे. Okay, you don't take it that seriously. But its importance is because you know wet lab very well. If you know bioinformatics also, that can be a very big thing add on to your resume. Okay, and the company or any research institute where you're going in, you do not it will be easy for you to understand and analyze the data. Yes, there can be some times where you need to collaborate with bioinformatician because some data is too tricky to understand or analyze. Okay, but whatever they have sent, to maybe to samajhna chahiye. All right. So this is where bioinformatics, having the knowledge of bioinformatics, plays a very important role.
Madhura, Madhura, you had a question. You can.
Yeah, sir. I had a question. So, recently I heard that DNA can also be the information that DNA or DNA itself can be used as a platform for coding, because you have a lot of 3 billion base pairs and all that. So how does that work? I was quite curious about it.
You mean to say you want to decode the information which is present in the DNA? Is that what you mean?
Yes, and kind of I had read in a newsletter that DNA can be used for coding because it has a lot of 3 billion base pairs, so it can be used. How does that work? I wanted to ask.
Okay, I need to look at the article, because DNA can be used for coding. In what aspect are you saying? But one thing I can tell you is decoding of the DNA is what is next generation sequencing. So in that genomics section, which I showed you about NGS technique, so you have that entire DNA sequence, and you want to identify what the genes are. You want to identify what is the functions. Okay, from the genes, what is its annotation? What do they code for? Okay, what is the molecular functions? All of this can be decoded from that one sequence using next generation sequencing. It can also predict what could be the possible mutations which can happen in that particular sequence. All right, so. Yeah, so you can decode DNA using this thing, but using DNA as a coding, I don't know exactly. I'll have to need to understand what exactly it means. Yeah.
What you mean is something like the binary numbers 1 and 0 can be used to crypt, right? A message or a code. That's what you mean.
Yes,
so the DNA with 4 base pairs can also be used similarly to send a message or a code, right?
Yeah,
that's what she means, Dr. Bapat.
Yeah, that can be done.
Sorry, I was not able to elaborate.
No, it's okay. It's okay. Yeah, so application of obviously your entire coding is based on binary, but we have done similar thing of binary for our in chemoinformatics, there was one of the study where we wanted to analyze that the different compounds based on its toxicophoric and pharmacophoric. So there we had used machine learning to differentiate between the compounds, and the parameters what we had used was we had developed our own features which were binary based. So what you're talking about using ATGC as a binary thing, in that we had used the different chemical components, and we had given them, so there were only two chemical components, so we had given them binary numbers, and it could be, we were able to differentiate between cancer compounds and diabetic compounds based on the binary fingerprints. This is what we call as fingerprints actually. So binary fingerprints which were given to them. And the paper has been published already a long time back. But yeah, application of binary fingerprints for coding is done. Can be used in multiple. I don't know how in DNA, I have to read that article on what aspect is, but yes, we can use binary fingerprints.
So, sir, I had, these days quantum computing is also coming into the foray. So, how will that be, used in bioinformatics to, make analysis better?
So quantum computing is already applicable when it, so all your actually molecular dynamics is based on quantum computing. What is molecular dynamics is, here we are talking about how the two atoms which are there, okay, how they are interacting with each other. So when I'm saying interaction, that means it's not just a hydrogen bonding or something. It's like there are various energetics which are involved in that. Okay, and how the energy between the two atoms are there. How much close they come, how much far away they are, and what is their relationship. So those can be the water atoms. What is their relationship with the atoms which are there in your compound, with those which are present in your protein. So how they interact. So and with this we use something called as force fields. Okay, again, force field is a part of your quantum computing, and how actually all of them are interacting with each other, and based on that only this movement of the compound in the protein, that is what it takes place. Another way where quantum is used is to determine, as I said, the energetics between so there's something called as energy landscape computation. In energy landscape computation is where you want to identify what are the various energies, something like potential energy, let's say for basic. What is the potential energy of a particular class of proteins? So for that, I generate the energy landscape for all of those proteins. Okay, that will help you to determine that based on the energies the proteins can be classified into different classes. All right. So quantum computation has been used. Quantum mechanics has actually been used in bioinformatics a long time back, that is in your molecular docking. A recent one where we had used was DFT energies, that is density functional theories, if you have heard of. So there's one paper which we published there. We used this DFT energy to calculate the energetics between the two proteins, and we had made a software. It's available on source code actually. The best part about that is it gives us the similar energies what molecular dynamics can do, but molecular dynamics takes a very, very long time to do that. DFT does it very quickly. So yeah.
Thank you so much, sir. The talk was very informative, and as I said, you explained things very well. So any faculty or any teachers want to say anything or ask?
Others, actually we also have other faculties who have joined. We have Dr. Simran. In fact, she teaches bioinformatics theory to the students right now. We have Dr. Sonal also who is with us. So any questions from your end, guys? In fact,
it was actually very informative for me. Hi, Dr. Bapat. This is Dr. Simran here. Hi. Even I've worked in the field of computational biology using molecular dynamics. I also we worked on CD4 intrinsically disordered peptide of that, and we came up with the energy landscape. I worked with Dr. Murarka in Indian Institute of Science Education and Research, Bhopal. I did my master's thesis under him. So yeah, so it was, I could link to all of the information that you put here together.
Valuable time on a Sunday, almost, I would say, yeah, almost 2 hours, close to 2 hours now. So yeah, so over to you guys.
Thank you, ma'am. We have come to an end of this session. On behalf of the entire Science Club of Sunandan Divatia School of Science, I would like to take this opportunity to express our heartiest gratitude to our speaker for today, Dr. Sanket Bapat, for gracing us with his presence. Thank you, sir, for taking time out of your busy schedule and addressing us today. It was very enlightening and encouraging to hear you speak about the emerging field of bioinformatics and its role in today's world. I'm sure it helped a lot of us to understand better the role of technology in development of science research. Also, we would like to thank Dr. Purvi Bhatt and Gurudas Mane for their guidance in organizing this talk. Thank you, ma'am and sir, for your constant support and encouragement to us. We would also like to thank all the students and faculty who have joined us today for such an interesting talk. And we hope this helped them to understand the field of bioinformatics and the role it plays in the research and development of science. Once again, thank you, Dr. Sanket Bapat, sir, for joining us today on the occasion. Before we end the session, I would like to quote something by Edward Teller, who was a physicist, and I quote, "The science of today is the technology of tomorrow." So on this note, with the theme of being the future of AI, we would like to conclude today's session, 2021. Thank you everyone for joining us today. Before we close, can I switch on the videos for a group photograph?
Yeah. Just I would request everyone to switch on their videos so that we can have a quick picture.
Also, I would like to say thank you, Purvi ma'am. Again, once more, thank you, Purvi ma'am. Thank you to the Science Club team members for organizing this event. Thank you, sir, madam, who had joined this session. So thank you for giving this opportunity. Thank you very much.
Thank you. It was a pleasure. Thank you. So